So you made mammoth meatballs.
Yes, I did.
First of all, how?
Okay, so what we did is we took sheep stem cells and then we replaced a protein called myoglobin, which is a protein that carries oxygen in your muscle. That's why muscles are red. Why meat is red, right? Because it can carry oxygen. And this protein myoglobin we replaced with a mammoth one, and that makes up 85% of the protein of meat. So then we grew those stem cells, billions and billions of them, and then a cook made a big meatball out of it.
A cook. So you prepped it. You
Yep.
did everything.
Yep.
Spaghetti style.
Yeah, it was like mincemeat. Yes.
Was it eaten?
No, we were not allowed to eat it because our immune systems, of course, have not seen mammoth meat for 30, 40,000 years.
Yeah.
So we would like to test it out properly before taking that risk. But at the same time, we were there when it was barbecued, and it smelled delicious.
Really?
Oh my God. Umami, meaty, gamey. Like, I want to eat this, right? That's a kind of barbecue smell.
I love that. We're already eating the endangered species. But if we can just sort of like zoom out for a second, what is it? So it's a mammoth meatball.
Yep.
But what is it? What does that mean? So like a woolly mammoth.
Yes, it's basically made out of artificially grown muscle cells that we engineered to be mainly consisting out of this mammoth myoglobin protein. So it's about the size of a small football, and at the moment it's exhibited in a museum in the Netherlands, so that people can sort of gawk at it. But the whole point was of that whole exercise was to highlight that you can make meat without having to kill an animal. So in an ethical way. It's also it produces less CO2 and less waste, right? So it's an ethical and slowly becoming more economical way to produce meat for consumption. And at the same time, it was kind of a cool idea. We were working with a company called VOW, and their whole market idea was not to make chicken meat or beef or lamb, because people know exactly how that tastes. But they were producing a mixture of chicken and scallop. They were producing giraffe and lion. So
Mixtures.
Yes, mixtures. Just testing it out, right? Because that's the kind of meat, first of all, that people not normally eat. Secondly, you can never ethically and sustainably make meat like that. And thirdly, people have no idea how it tastes. So if it tastes a little bit different, people go like, yeah, that's probably how lion tastes like, right? So yeah, so that was the idea. So that was where the idea came from of making a mammoth meatball.
Right. Okay. So how far back, or how old or fresh does a species have to be for it to be, I guess, like viable in that process?
What you need is the DNA sequence.
Right. Okay.
Right? So we were lucky because this type of meat that we were making was 80% made out of this myoglobin protein. So we only needed to replace one protein with a mammoth protein to make 85% of the meat, mammoth. The rest was still sheep. Right?
Sheep.
So
Okay.
it was a sheep mammoth meatball in a way, right? But what was I saying? The DNA sequence of that myoglobin of the mammoth, we knew from these frozen mammoths that are found in Siberia. They sequenced the DNA so that we know exactly how that protein looks like in mammoths. And the bits that we don't know, we filled it up with a little bit of African elephant myoglobin bits of the protein, so that it still looks and acts like myoglobin. But the really amazing thing is that so myoglobin in a muscle cell, myoglobin organizes itself into fibers, right? Because it's your muscles need to contract, right? You can imagine that those are muscle fibers. And the mammoth myoglobin happily integrated with the sheep myoglobin and played together nicely in the sandpit with the sheep myoglobin and made fine muscle cells. So that was kind of a surprise. We didn't know that, right? What happens if you re-express an extinct protein in a cell of a sheep that's walking around now? You have no idea what's going to happen. Maybe the cell's just going to go like, well, we don't know this, and it kills itself, or it dies, or it doesn't work at all as myoglobin. But it just still works. It also tells you that even species that are really not here anymore for a long time, the basic biology principles are kind of the same, because we evolved from these guys. So this kind of makes sense. So and so that was a really interesting project, because it sort of sat at the interface between educating people about genome engineering, genetically modified foods. That's clearly a genetically modified food. And sustainability and making people aware that we need to change our ways if we want to save this planet, right? Mm-hmm. So that's why we were super happy to be involved in that. Yeah. And we are just about to launch a second project with a different company now, a leather company, an artificial leather company, because leather is still made by killing animals, right? But you can also make skin cells out of stem cells, and instead of muscle cells. So now you can make leather, and that's what this company does. So now what we've gone a 35 million, well, a little bit more than that, more than 40 million years earlier, because people have now found little bits of a protein from T-Rex.
Hold on.
A collagen.
You're about to get me excited. I need to brace myself. Okay, continue.
So people have identified bits of fossilized T-Rex muscle, and from how the fibers of the fossilized fibers looked like, they could see that this was a particular protein called collagen. And collagen is what is excreted by your cells and glues all your cells together in a way, right? So the glue between your cells. And that also gives skin its strength, right? And its properties. It's supple, it can move, but it's tough, right? So this particular collagen was found in these fossils, and they also found a little bit of DNA, and they were able to get little bits of this T-Rex collagen. And then we took those sequences, so a protein is made out of amino acids, right? By a sequence of amino acids. It's basically like beads on a string. And we have the little bits of beads we have of the T-Rex we had, but we had huge holes in that protein, of course, because you don't find a lot of T-Rex collagen after such a long time.
No.
So we basically replicated that little bit, and we did a little bit of AI comparison of other dinosaurs collagens that were found and how they looked like, and then we asked AI to come up with candidates, and then we selected the best ones. And now at the moment we're expressing this T-Rex collagen in a skin cell, and that's going to be used then to make leather that is going to be, at least for a substantial part, made out of T-Rex collagen.
Can I just ask a quick question here? And benchmarking these projects against, I'm sure at the time everybody was asking you about the dire wolf thing.
Yeah.
And I know Joe and I have discussed this, and I'm vague on the details now, but I remember at the time reading some of the commentary and listening to interviews and going, this is a marketing stunt, because based on the way they've described it, I'm of the firm opinion that that is not a dire wolf.
You're correct.
The way they've broken it down, and I'm like, no.
You're correct. Yes.
So whereas this is actually involving sourcing primary material.
Well, so was the direwolf, though.
So was it.
Can you explain the difference between the two?
So, to my understanding, what they did is they made genome edits, right, in a normal wolf to give it dire wolf features.
Oh,
yes,
right,
yeah.
So they took an existing wolf and the eggs probably of an existing wolf, and then they made these particular mutations and genetic changes that are found or were found in dire wolves, and then dire wolfed a real wolf in a way, right? And so that, and in a way that's exactly sort of what we have done. We've done with the mammoth meatball. As well as with the T-Rex. It's kind of similar, right? Because not the entire organism is T-Rex, or the entire bag is not T-Rex. The entire meatball is not mammoth. There's another cell involved, right? In one case, a sheep stem cell, in the other case, a chicken skin cell. So if you see the dire wolf as the skin cell, right, or the normal wolf as the skin cell, and you then mess with the genome and you change the genes in such a way that they start to look like like dire wolf genes, then what you get out on the offspring is going to look like a dire wolf, and that's exactly what they saw. So it's kind of what we did, but then in a living organism, I guess. Yeah, but it's not a dire wolf, right? Mm-hmm. A dire wolf would be. And a real mammoth would be if you would take a cell from one of those frozen mammoths, right, that has been frozen for 30,000 years, and you would be able to revive it in a dish, and and you could make it grow, or you could take the nucleus where all the DNA genetic information lives, and you could take that out and put it, for example, in a skin cell of a mammoth, or no, of an elephant, I should say, of an elephant. So now you have the cell of an elephant, but now all of the DNA will come from the mammoth that was frozen in the tundra in Siberia. Now, if you would reprogram that cell back to a stem cell or to an egg and a sperm, or you could make, and you would implant that artificially created embryo in an elephant or elephants, you probably need hundreds of them, then you might get one real mammoth out on the other side, right? So that's the difference between messing with the DNA of an existing species to make it look more like a mammoth or a dire wolf, or taking all of the DNA of a mammoth and making an entirely new organism out of that.
Right. Is that at the moment, is that theoretically possible, or is that possible right now?
No, that's not possible. That's possible.
And this is crazy because we've gotten very granular very quickly, but Jurassic Park, which was one of my favorite movies, that's possible.
Yep, that is possible.
Whoa, hold on. It's possible for certain species, like species that have been preserved well, or it's possible for anything that we can find some level of something. So like a T-Rex collagen, we could make a huge chicken that would look like a T-Rex, and we could edit the DNA of the chicken and we get a T-Rexy looking thing.
But you see, you've identified the problem here, right? Right? Is that a chicken is not as big as a T-Rex.
Right? These little jacked kids walking around flipping cars.
I can hear KFC knocking at the door, going like, you get some pretty big drumsticks out of those. No, but that is exactly the problem with mammoths and with T-Rexes and other big species, right? There's nobody to carry these things to term. I mean, a T-Rex is probably an egg, right? But at the same time, to change a chicken to a T-Rex, that would be absolutely impossible. There's too many genes that are different, right? So to de-extinct a T-Rex, you would need, like in Jurassic Park, you would need to get
frog
a blood cell that is totally from the T-Rex, and then you would have to make sure you would have to be super lucky that all the DNA is still intact. Then you can either sequence it and synthesize it, that you can do it now these days, or take that DNA and put it in a fresh cell, and then now you have captured the DNA from the T-Rex. Now you need to start making eggs and sperm, or to make an artificial embryo out of that. Now you need to somehow find a way to keep that embryo alive outside of a T-Rex environment or outside of an animal like a mammoth. But to tell you the truth, I don't know how T-Rexes were raising their kids. I suspect it's like all these lizards, right? They just laid eggs and they hatch and then they're on their own, right? So maybe with a T-Rex eventually it would be possible to just to make eggs and hatch them.
No way.
Yeah. If you would have intact DNA from any species, you can do that. Yes.
Well, I think that would silence the dinosaur non-believers. If you're like, nah, I still don't believe. You're like,
I don't know what you made. No, that's a monster. That's wild. Actually, interesting question. The Jurassic Park theme, because that's all really, really cool. But I think where people kind of get tied up in knots is people spent so much time trying to figure out whether or not they could, that they didn't stop to consider whether or not they should.
Yeah.
And so going back to, and this sort of ties us back to the start of this and that mammoth meatball, and you touched on, I guess, some of the social functions that something like that can serve. But what is behind, what is motivating this sort of research? So I guess there's the scientific innovation and ingenuity, but in terms of like the social driving force behind it, what is it?
Yeah, for bringing back to de-extinct species, for species like dodo, I can sort of understand it, right? Because one, they're not gone that long, and secondly, they probably had a function in our ecosystem because they died out 150 years ago.
It's deforestation, was it? Yeah.
Well, I know that they lived on an island and they just basically killed them all. So I think that will be all right to do, but to bring back a species that is extinct for a reason. First of all, while we are totally destroying our own ecosystem at the moment, seems to be like a waste of resources to me, and I think to most people. So these projects, some of them one of them is funded by one of the big oligarchs in Russia, I believe, that spent a lot of money to de-extinct the mammoths because he likes the idea to have Russian tundras with mammoths again.
That's so Jurassic Park.
So that's sort of the level of that. Serious scientists are not looking to de-extinct species. They are much more interested in using the same technologies to improve health outcomes for people, or to produce more ethical meat, or to
Mammoth meatballs.
Or mammoth meatball, or as I said, a mixture of quail and chicken. I mean, it's absolutely delicious, right? So in an Earth where there is a part of our population that eats way too much meat for our own good, when we could save this planet by actually turning mainly vegetarian, right? that's going to save us. I think to produce artificial meat for people that really need to have meat without killing animals and without stuffing the climate of our planet up, I think is super relevant. And that's this other stuff that we're talking about is a little bit more peripheral to that, right? But it's these flow-on effects that are enabled by scientists seriously looking at that angle, rather than at de-extincting, an extinct species for a billionaire. That's not what it's about.
That is interesting because like I am an absolute carnivore, but at the same time, Joe and I have had discussions about basically the scale of the pigging or the chickening, and you go, do you actually need massive warehouses where it's just all about the scale of the produce that goes through there? And
that.
It's ethical questions over that. And you go, do you need it?
There's that aspect, but there's also it's just a resource question. Right? I mean, I forgot what the exact number is. I think 30% or something like that of what a cow eats is turned into meat, and the rest goes into methane and walking around and doing cow things, right? So if you think about it, if you could use the same vegetable resources that the cow eats and actually eat it yourself, you get 100% of the resources out of that, instead of giving it to a cow first and then only get 30% back. And you have to deal with the waste products, and you have to deal with the ethical issues, and you have to have room for them, and you have to look after them. Maybe it's just eventually on a planet with billions and billions of people, that are more economical ways to feed people, rather than giving our corn to the cows and then eat the cows. Like, just eat the corn, right?
Right.
So from that point of view, I think that's something that as a planet we need to change our ways a little bit. But as you say, people have been meat eaters for evidently 40,000 years, because allegedly we hunted, or we and the Neanderthals hunted the mammoth to extinction, probably because they tasted so good, right? so there's a reason why we like meat, but one, we eat way too much meat, and secondly, if we can produce meat in a more sustainable fashion without cruelty, I think we should seriously look into that, right?
It's interesting that you say that particularly, because I think in places like Africa, a lot of their economy relies on what do they call it? Guided safari hunts, and the hunts tend to be good for the ecosystem because when you're shooting, essentially the idea of a trophy hunt is that you pick the oldest male out of the population and you will limit him for, obviously for the meat, etc. But what that does is, if in the case that male is too old, but he's extremely dominant and infertile, he can kick all the young suitors out of the squad, and then you have a bunch of females chasing a unpropagative male around the place that end up, dying out. So eliminating him allows the younger, I guess, more viable suitors to go and help breed and boost that population.
Sure, Joe. I
No, I'm not.
I hear you there, but there are thousands of trophy hunters that want to do that, and there are maybe a couple of thousand lions left, right? So the numbers just don't stack up at the moment.
No. And I'm not talking specifically about any sort of species or the problem with hunting in general, but the idea behind that specifically, which is why America versus Australia in terms of the culture around hunting. In America, you have to have a tag, and so in some states, certain species you're not allowed to shoot them because they're endangered. But then in other states, they've got a problem where farmers and agriculture so they need hunters to go there. But like I'm not. This isn't me trying to make a case for hunting because I understand the problem with it in that aspect, but if you could create meat in...
Can I jump in and ask a question? Do we need meat? Because I've had this discussion with people, and you go back and you say, I've heard some people make that argument, and you go back and we're apparently, and I'm willing to believe that we've eaten meat for a very, very long time, and our modern diet is very much centered around, accessing protein from certain things. But I think you just said that we don't. Sorry, this may not have been captured on the audio. But you said we don't need meat.
You don't. No, there are people that. My own daughter is vegetarian now, vegan, right? So yeah, she doesn't eat any meat. She's alive and healthy and well.
Yeah.
And I've started to eat less meat. My cholesterol is better. And yeah, I haven't totally given up meat, but I'll eat a lot less meat. But yeah, you don't need meat to survive. I mean, there's a whole continent like in India, right? Most people are vegetarians.
Right.
They are clearly doing fine. Yeah.
So just to almost steer back to the beginning, where I guess the origin of this whole conversation comes back to stem cells.
Yep.
And what you specifically do, because we're tossing out big ideas and huge projects that are epic to talk about, but we haven't even
explained anything.
We haven't. Like, I'm still going. So how is this possible? So what are stem cells, and then go from there.
I think that's a sensible thing to do to talk about a couple of minutes. So a stem cell is a cell that has two properties. One, it can make a copy of itself, or it can turn into a different cell type. Which is not the same, has a different function and a different appearance than the cell that it used to be, and that's a process called differentiation. So it can copy itself, so you can make more stem cells. That's one choice it has, or it has a choice to become a different cell. And in our body, when we are a day 5 embryo. Just before you were a day 5 embryo, you're just a clump of stem cells. And then there's a process called gastrulation where the first 3 different stem cells are made. So and that's, sorry, they have different terms. They're mesoderm, endoderm, and ectoderm. Basically, mesoderm are the stem cells that are going to make your blood, your heart, and things like that. Then you have the ectoderm that will make your skin and your brain. And then you've got the endoderm that will make things like lungs and your intestines, for example. So you can see that different, really early on at day 5, when you were a day 5 embryo, at the day that you implanted in the placenta of your mom, at that stage, then over the next 24 hours, those 3 stem cells are made. And that's enough to make it an entire human being. So those stem cells that are grown before they become these specialized stem cells, they are called pluripotent stem cells. Pluripotent means they have multiple potencies. They can make all the cell types of the human body. And that's the first important step in creating a life. And this happens in all of the mammals, for example, right? And this is how you make an embryo. So that's one type of stem cells that I have been working on my entire life. And then there are stem cells that come from these stem cells that are sitting in your body at the moment. These are the stem cells that sit at the basal layer of your skin and that sit in your liver and that also sit in your brain still a little bit, that continuously make new cells. Because you must have noticed that you are continuously losing skin and cells,
and
right?
hair.
And hair and everything. But unless you get older, we can talk about that later, those things usually grow back, right? Because every time you look in the mirror in the morning, you kind of look the same. But lots of cells, thousands of cells have died. Thousands of cells are dying in your body right now, but you just don't realize it.
It's bizarre.
Yeah. So they need to be replenished. They need to be replaced by new cells. And those stem cells that are sitting in our different tissues, they have the job to make more of those cells. So evidently, those stem cells that are sitting in your liver, they don't make brain cells. They make only liver cells. And vice versa, cells that sit in your brain don't make liver cells. So they have locked into very specialized roles. But all of those stem cells that are still sitting in our bodies, they come from those 3 stem cells that I talked about a couple of minutes ago at day 5 of development, these pluripotent stem cells. So they are to me, they were the most interesting stem cells to work on because they will grow forever. So you can make unlimited amount of these cells and they can make all the cell types of the human body, which means that we can study how a heart is built. We can study how a heart development goes wrong when you have a genetic mutation. You can look at the impact of viruses on the brain without having to take a piece of somebody's brain out. So it has opened up the ability to make different cells from different tissues has really changed the way that we can do research in medical research.
And what something I found really fascinating about that, I did do a little bit of homework for this conversation, was distinguishing between, I guess if you'd call embryonic stem cell research, and that's a can of worms for some people on ethical grounds. But then there's another term that I think you give to basically, I don't want to call it reverse engineering stem cells, but almost taking, so probably what you were talking about.
Correct.
Then something that's much more progressed, taking it that progressed stage and then reverse engineering it down to those fundamental building blocks, but that you can then work from.
You have done your homework. Well done. Yes. So those pluripotent stem cells that I was talking about originally, and we're now talking about the end of the 1990s. Originally, they were isolated from real human embryos that were donated to research, surplus IVF embryos. You know how people do IVF and to get, because they're infertile, they implant an embryo and then they have 5 embryos left. They donated those to research, and there, clearly, you can isolate those stem cells from those really early embryos because they haven't implanted in their mum yet. So that's where these embryonic stem cells came from. And then clearly, you're destroying an embryo to get those cells growing in your dish. So there are clearly, for some people, there are ethical and moral questions that need to be asked. That changed when in 2007...
It's a Japanese guy, wasn't it?
Yeah, in 2007, he came actually to Cairns, to one of the big conferences, and I was there. And he presented the data that he had done this in mice, where he could take a skin cell from a mouse So just by taking a small snip of the skin, and we can now do it with blood cells, cells that are shed in the urine, any cell that will grow in a dish. But he showed it with skin cells. He showed that you were able to bring those back to that day 5 embryo stage, all the way back down to development where you're a day 5 embryo, and make those pluripotent stem cells without using an embryo, but just forcing them back in a cellular time machine, if you want. And he did that by delivering powerful transcription factors, basically master control switches that force the cell back into its state, even though it's not something that it normally does. So it basically changed the whole makeup of the cell to make these what's now called induced pluripotent stem cells. So they're still pluripotent stem cells, but they don't come from an embryo. They were artificially made, as you correctly pointed out. That's why they're called induced pluripotent stem cells.
So what was the science world's reaction to that when you, like all of you listening to this?
We were gobsmacked. I mean,
Yeah,
this was one of those seminars that you will remember for the rest of your life, because it was packed. People were hanging off the rafters, and Shinya Manaka, that was his name, was telling this story how he had done this, and everybody was just gobsmacked, because everybody was working on these embryonic stem cells. So were we, but when he showed that this was possible, and everybody immediately realized, okay, if you can do this in a mouse, you can do this in a human cell as well. And he showed a year later that you can do that in human cells as well. But everybody sort of knew that at that stage, everybody who was in the room. But what it does is it now allows you to look at an individual and say like, oh, okay, you have this particular disease, right? Let's say a genetic disease. And I can now take a little bit of your blood or your skin, and now I can make those induced pluripotent stem cells from you, which has your DNA and your mutations. And now I can start to learn how that leads to disease, and then start to use those cells to test novel therapeutics and drugs to combat the disease. And that, of course, you capture the individual makeup of the mouse or of the individual that you reprogram the cells from.
So is that almost like, I don't want to get the terminology wrong, but almost like you've got a differently formed avatar of yourself that you can test things on.
Exactly.
And then if it works, and that's a question, I guess. Hang on, this is how we come into organoids?
Yep, correct.
And what I'm really interested to dig down into is what the heck is an organoid?
Right.
And what about an organoid is transferable and sort of, I guess, I don't know if what word? Congruous.
How good are they?
Yeah, and comparing it to its human.
Yeah.
If you put it that way.
Yep.
Or can you compare it to its human? Like what is an organoid?
Yes. So just to lead on from what I just said, you can reprogram any cell of an individual into these pluripotent stem cells. So what everybody started to do is to make liver cells out of them, or heart cells, and grow them, as you see always on the TV, in these dishes in 2 dimensions, right? But everybody knows that a heart or a brain doesn't work in 2 dimensions. They are 3-dimensional interconnected networks of cells that work together to give it the function of the organ that they come from, right? So that's what organoids are. Basically, these stem cells, because these stem cells build the different organs as we develop, right? I told you that these pluripotent stem cells make all the cell types of the human body. So they build your heart, your liver, your kidney, your brain, your bones, your skin. They build everything. And they do that because these stem cells have a program built into them to make that particular organ. And what you do is, by balling these stem cells up, you can take these stem cells and put them on the path, let's say, to a brain, and then allow them to do their own thing. Allow them to imagine that they are a particular part of the brain. You give them the right environment, the right growth factors, the right, you're looking after them as to fool them that they are thinking that they are in a brain environment. And under those conditions, those stem cells know nothing else than to build what they normally build. So they build a little bit of brain. And that can be done with now every organ of the human body. So you can build little heart organoids that are beating with the frequency of a human heart. You can have little kidney organoids that are producing little bits of urine. You can have liver organoids that are detoxifying alcohol in the same way as our own liver. And you have little brain organoids that are demonstrably doing data processing and doing all the things that brains do. So that's where organoids have become really powerful models, because not only do they have the cells and the cell makeup of the organoid in question in the right proportions, because that's what stem cells do when they build an organ, but they also have at least some of the functions of that particular organ. And that is basically what defines what an organoid is.
So definitionally, what's the difference between that and like cloning?
So cloning is when you make an entire organism with the same genetic makeup as you. If I want to clone Joe, right? But the organoids still have. If I make the stem cells from you, the organoids would still have your genetic makeup, but they wouldn't be clones, right? Because they're not a whole Joe, right? They're not an integrated. They're not Joe.
Yep.
So if you want to clone somebody, you have two ways you can do it, right? So one, you can take those stem cells we were talking about. They can make everything in the human body. So they can also make sperm, and they can also make eggs, right? Like we do in humans and females. And you can then, for example, make artificial embryos out of those, right? And so now you have cloned Joe, right? Or you could take a skin cell from somebody else or from your brother, right? And take the nucleus out, right? And now replace it with your DNA. Now grow lots and lots of those cells up, and then reprogram them again and do the whole exercise again. Or you can take the nucleus out of one of your cells and directly implant it into an egg. And then make an embryo from that.
Right. So you still need the kind of.
You still need an oocyte, yes,
right.
an egg cell, and that's how they clone Dolly, right? So for
Dolly the sheep.
Dolly
I
the
haven't
sheep.
heard that name in a long time. And hang on, I'll let you finish, but I want to ask before I forget, what's the status of cloning? Because you used to hear about it all the time, and I guess now you're doing
old
organoids and chimeras, and so we're probably like way past cloning. But like, what's the status of the cloning world?
Well, nobody is cloning anybody, right?
Or we just don't know about it.
Or we don't know about it. I mean, there was this whole So one, it's against the law. Secondly, it is exceedingly difficult because there's lots and lots of failures, right? Even with Dolly, I forgot how many, but I had to do dozens and dozens of sheep to get one that had a healthy offspring.
And she died, right?
Yeah, eventually she died. Yes. But you can see that in humans, you can't have 50 or 60 women that are made pseudo pregnant and then become recipients of some kind of clone, and then all of them birth monsters, and one of might maybe a real clone. It's unethical and nobody's going to go for it. So and finally, I don't think we have found anybody worthy of cloning yet, right?
Not in this room, at least. Maybe you.
No,
maybe
so it's exceedingly difficult. It is not worth doing, and there are much better things to do with our time. Now, having said that, where cloning can become really useful is, for example, in livestock production, right? And we had a program with a company here in Queensland. To do what we called artificial twinning of embryos, of beef, of bovine embryos, of cows. Because if you think about it, up in the territory we have what 200,000 herd cows running around, and when it's the time, 10 or 20 bulls are let loose and they do their thing, and then you select the best ones out of that, and 30 years later you may have a better breed of cow, right? Because it just takes that many generations to drive new genetics through a population. But if you could select your best bull and either inseminate all the cows with that bull, or take your best cow and just make 10,000 copies of that bull and insert those embryos into the cows. Now you have 10,000 cows with all the same genetic offspring of your best bull, and you have the better genetics the next year.
And taking it on that version of events, is that actually how it works? So like in theory, you would want like the archetype to be propagated across, but is that you don't lose something in the propagation?
Not necessarily. I would think no. I don't see why you would.
Right.
No, but what you lose is diversity, right?
Yeah.
That's what you lose, right? But over the last 100 years, as we've been breeding better cows, we've been selecting, better cows and mating the best ones with the best ones, and now you've got
Super
bigger
cows.
cows with more meat and more milk production. The natural way, which is the slow way. The fast way would be to just propagate the good ones, right? So that would be the same cows. The problem might arise when there's now a virus infection, right? That infects the Northern Territory. Now all of those cows will have the same immune system because they're all genetically identical. So one virus might just wipe out all the cows in one go. And that's what you wouldn't happen when you do it the natural way, because now you still have some of the genetics slowly going through through your population and becoming better, but they're all a little bit different from each other, right? Because they all have different moms and different dads and different sperm. One sperm is not the same as the other. So you have diversity, and that creates resilience in the population, right? Yep. So it's finding this balance, right? And this will be a theme that will come back in most of our conversations. It's like you can see that you can do good with this, but on the other hand, you don't want to push it too far because you might be doing things that you didn't intend to, right?
Yeah. And when you say good, and I'm sure this is it always seems to be looked at in through the lens of like commercial optimization. So there are other forms of good, but that seems to be when people get into trouble, when it's sort of trying to optimize nature out of it. And based on what you've said,
yes.
You sort of do that at your own peril.
Yes, this idea was picked up and funded by the Gates Foundation to be introduced primarily into a country in Africa and in India, where cows are so rare and so valuable, right? Is that they don't have the luxury of breeding for 30 years to improve their meat production, and they definitely need more meat. So again, you might want to do a little bit of that in India to really allow these people to live healthily, because there they don't have enough meat. But on the other, you don't want to push it too far so that you lose your diversity, right? Mm-hmm. So yes, there's always an economic imperative, and the problem is that many of the politicians and the policymakers are not aware of the intricacies, and this is why scientists like us are going on podcasts like these to tell people what the deal is. Right?
This podcast,
this one,
this podcast.
So you specifically would you say specialize in brain organoids?
At the moment, my lab,
Yeah. So with the brain organoids, what are you researching, and is there any sort of groundbreaking stuff that has come up as a part of your research?
Oh, geez, so many things, Joe.
Oh, sweet. We've got all the time.
I mean, I must say, I have started to fall in love a little bit with brain organoids for a number of reasons. So the first one is that brain organoids are a little bit like the Mariana Trough, right? I like in the seas. We've been there maybe 2 or 3 times, but we know bugger all what's happening in the seas, right? I mean, we've been to the moon, but we don't know what happens in our seas. And our brain is a little bit like that. We know it's really complex. We know it's amazing, can do amazing things. But how it actually works, why we sleep, what consciousness is, how are memories stored, how is computation done in the brain. We're pretty smart, but we don't know any of those answers of any of those questions, which is kind of insane, right? If you think about it, 150 years of brain research, and we still don't know those fundamental questions. So one of the reasons for that is that because people are kind of attached to their brain cells, right? Because if I was a liver researcher, I would say like, Joe, I just need to stick a needle in, on the local. You might not like it, but I need a bit of your liver to tell whether you've got a liver disease, right?
Yep.
You go like, look, I don't like it, but I'm okay. If I have to, I will, right?
Yeah, yeah.
But I want to ask you, now you got a brain disease. Can I just grab a little bit of your brain because I just want to study it in a lab? Are you okay with that?
Sounds aggressive.
Hang on, could you do that and not screw him up? Wouldn't
No.
that be like a lobotomy?
Yeah, exactly.
Right, okay.
I'm like, what am I missing here? All right.
That's exactly the thing, right?
Yep.
I mean, because you can't do that without penalty.
Joe would lose all his personality.
Which would be a world disaster.
Such a crime.
No, but seriously, this is the problem. Why it's very difficult to study real human live brain cells because there's no access to it, right? People don't want to donate that. So this is where brain organoids come into their own. Because I can make stem cells from anybody, you guys, myself, anybody with a particular disease via this reprogramming method that I told you. So now it has your genetic makeup, and now I can make thousands of brain organoids that are the same as the front part of your brain, the hind bit of your brain, the mid part of your brain. I can connect those bits up with each other. They start to talk to each other, and they have the cells and the makeup and the behavior of brain cells as we know that they should behave. So that means that I now have a little insight into how a little bit of your cortex might be working, right? A little bit of Joe's cortex. Now, God forbid, let's say that you have epilepsy, right? Most people know somebody with epilepsy. And it's a terrible disease. You can't drive, you can't do anything because it can hit you anytime, right? So it's a very debilitating disease. Now, one of the things that we've done that with people that have epilepsy, and we made these brain organoids. So the first thing that we see is that these brain organoids have abnormal activity. Sometimes they just go nuts, right? They have an epileptic attack. They have hyperactivity of the brain. They do that without us telling them to do. So now we can say like, okay, which of the existing drugs would be effective in treating this particular individual, right? So now we can start testing our drugs out on those brain organoids and see which one of the drugs or which combination of drugs is most effective in reducing that epileptic activity of your brain organoid, right? Of your brain organoid.
Yep.
And this is basically one of the programs that we did, is then we take that information and we test it out in the clinic to see if our predictions are right. And we're getting pretty good at that. So that's one way. That's what's called personalized medicine, right? It doesn't get more personalized than that.
And can I just ask you on that? Because the personalized nature of it, is there a difference? So you're testing it at point A on an organoid, and then if the test is successful, that test is then done on a human,
Yep.
hypothetically. But the difference between an organoid and a human, and looking at like a brain organoid, very specific in isolation that from everything else that makes up a human, are there any obstacles?
Yes.
That sort of, how does that work?
Yes, I mean, obviously we can't build a brain-sized organoid in a dish yet, right? We might in the future once we give it a blood supply and we can make it big enough. But at the moment they're pretty small, so it's evident that it cannot be doing all the functions that our brain does, right? The other thing that it doesn't have is it doesn't have a connection to all of the other organs, right? It doesn't have a connection to the liver and to the blood vessels and to all the other hormones and things that are floating around in our body, right? So the way to solve that, or how it's being solved that at the moment, is to make different organoids of the different organs of an individual and then connecting them sort of with pipes. Right, so with an artificial blood supply. So yeah, this is called organ on a chip. So then what you can do is you can give your drug and pretend it goes into the gut by giving it first to the gut organoid. Then from the gut organoid, it goes to the liver. And then from the liver, just like in your body, it will go into your bloodstream and then it goes to the brain organoid. And then we'll see if the drug still works.
Forget agentic AI. This is so much cooler. So it's like a closed-off organoid system that mimics.
Yeah, a body on a chip.
Yeah.
So this is where it's now and where it's going. And this is also one of the reasons why our company is hopefully is going to be successful, because you need organoids for that, right? So that's one way you can tackle that. The other way you can do it is to start physically connecting different organoids with each other. For example, in the brain, we can now connect the spinal cord. We can make spinal cord organoids and connect them to your hindbrain and then to your forebrain, and really make almost an entire central nervous system that now starts to talk to each other and interact with each other like it would. So like a mini version of your spinal cord and brain.
And what is that? So if you've got a brain organoid and a heart organoid and a liver organoid, and let's say hypothetically an organoid of everything that can, the building blocks of a human body, but the status of that overall thing, it's not a human.
No.
Question. Okay, I would think that. What is it? Is it literally just a network of organoids?
Yes, it's a network. It's a network of little bits of you, or that's grow in a dish. Yes. So it's you could equate it to giving a blood sample, right? And then people do different tests on your blood sample. Your blood is still alive, right? Yeah, a blood culture is still alive. In this case, it's just like a little bit more complex and like a real bits of tissue that you can recognize, and which makes it a little bit more icky, I guess. But to me, there is very little ethical issues there, right? Because none of those organoids can ever propagate. It cannot become a human. It cannot become a clone. It is totally dependent on us feeding it every day. It can't survive by itself. So there's really the only ethical issue associated with that. It is super you, right? It is hyper you. So this is one of the issues that we often have in our research, is that for rare diseases, for example, we know all the patients. We give them different numbers where everybody kind of knows, like, number one was Annie, right?
Right. Yeah.
But we pretend that we don't know. But this is the problem with these personalized models. They are personalized models, right? So sometimes you can find out things in a personalized model that you didn't want to know, right? Let's say that your brain organoid starts to develop signs of Alzheimer's disease, which it can do, right? We've made brain organoids from people with Down syndrome, who all get Alzheimer's disease at about 45 years old, all of them. So when we do this in the dish, what do we see? We see Alzheimer's disease in the dish, in Down syndrome brain organoids. When we look at brain organoids from kids with hypomyelinating disease, which is the disease where the insulation around your nerve doesn't work properly, because they're just like electrical cables on nerves, right? They need to be insulated. If that doesn't happen properly, you have serious neurological disease. So we do that. And what we see in our brain organoid, we see less of that insulation being deposited, and we see exactly the same things that we see in the brains of these kids.
And can I just to illustrate that? So let's say you do an organoid of me.
Yep.
You take my stem cells.
Yep.
You reverse engineer.
I don't
them.
even take your stem cells, just a blood sample.
You take a blood sample, reverse engineer it. You've got a Tom organoid.
Yep.
I don't have to have Down syndrome, but you can analyze my organoid and see whether or not there are telltale signs of Down syndrome in my potency.
No.
What does that look like?
Because Down syndrome, we know where this comes from. Down syndrome comes from an extra chromosome 21.
Okay.
And we're pretty clear that you don't have an extra chromosome 21.
But that was just an example. Like the idea being, is that how it works?
But if you had a disease, yes. Let's, for example, if somebody in your family has Parkinson's disease, an uncle and a nephew, right, it might run in the family, right?
Right.
So there are two ways you can do it. You can take your DNA and sequence it and see if you have Parkinson's disease mutations, or you can make a model like that, a brain organoid model, and then see if your brain organoids get Parkinson's disease. And the advantage is that once they do, then you can start to find out which drugs will work for you best.
Right.
Having to take them yourself first.
Is this in a testing phase, or is this something that's, I guess, open to the general public?
Oh,
So this is what most of the big pharmaceutical companies are doing now, right? So all of the big pharmaceutical companies have set up major organoid facilities to make organoids to test their drugs. So this is not mainstream in the hospital setting as yet, but this is something that we are advocating for very strongly, that it should be part of our general healthcare approach, because it can save us billions of dollars of money, right?
Yeah.
So that's, I mean, decades ago, this was one of the ideas that I had, is that you could take cord blood, right? So you know what the umbilical cord is, right? So normally when a baby is born, in the umbilical cord still has stem cells also from the baby. It has stem cells from the mom, and then also has stem cells from the baby. And they're the best stem cells ever. They have never seen smoke, ionizing radiation, alcohol, anything, unless the mom drank, right? But they like the most pristine stem cells you can imagine, right? You normally at the moment you can store these because you can make blood cells out of these, so that you can cure leukemia if you have it in your kids. But why don't we store the cord blood for all the kids in Australia as they are born, right? And then reprogram them into these induced pluripotent stem cells. Now you have a lifelong repair kit sitting there for each individual in Australia. And secondly, you would have the ability to start to screen drugs for whatever disease they may be getting later. You can immediately test which drugs are best for them.
Right. So what's the hurdle? What's stopping us from doing that?
Money and political will. Yep.
Right. Okay.
Is there any information privacy concern of scale that plays into, like a lot of
that would be
the utility that you're conveying? You go, oh, that's serious stuff, but. And I'm not even sure, like you can manipulate everything at the end of the day, even if it's like got incredible social utility. You can always flip it upside down. But what are the main ethical considerations that people talk about in this world?
So in this world, the first one is, at the moment, things that are happening in the States already, is that employers want to have your DNA sequence to find out if you have any genetic diseases, right? Or if you have a high chance of getting a heart attack or Alzheimer's disease. Insurance
companies.
So that's clearly unethical. In Australia, this is not allowed, right? But there, it's already becoming part of of the selection process. So that's clearly a problem, because your cells will have your DNA. So and your DNA is your property in a way, right? But on the other hand, you can find out bad things, but you can also find out that you are unlikely to get Alzheimer's disease. So some say, like, I don't want to know. Most people say, I don't want to know. I personally would want to know, because I want to be able to adjust my lifestyle and to minimize the chances. So but many people say, I just don't want to know. So this is one of the issues, right? Mm-hmm. Is that people would like to be ostriches and sort of let things, happen as they happen. But the biggest problem is just political will and money to get something like that out. The ethics can be handled. It needs to be surrounded by really good security. It needs to be multiple separations between the you don't want people access to everybody's DNA sequences. Yeah. But on the other hand, I can see that if we're doing horizon scanning, this is something that in 50 years is just going to be par for the course, right? In 50 years, you will have a motorcycle accident. You will tap into your stored stem cells, make some motor neurons, inject them back into your spinal cord, and fix the problem. Wow. Right? That's happening now with Parkinson's already, right? Oh, right. There are clinical trials going on now for people with Parkinson's, where there's a specific death of a specific type of neuron in your brain called the substantia nigra dopaminergic neuron. Those you can make from stem cells, and you can now make them from these induced pluripotent stem cells from you, so that when you make those cells and you inject them back at the right spot in the brain, your immune system will not reject them because they are your cells, right? So it will just accept it as if it's yours, and they seem to be working pretty well in animal and primate studies, and that's why now the first clinical trials are happening with that for people with Parkinson's. So this is not longer science fiction. This is happening right now.
What about the IP? So you're working on my organoid. Who owns my organoid?
Well,
Do I sign a waiver that gives you the...
in many cases you sign a waiver that you do not have you will not get financial benefits from that. But on the other hand, you also always have the right to withdraw yourself from the research and demand that your cells be destroyed. So you're both in control and you're not in control, right?
So that actually brings into account into conversation Harriet
Lacks.
which is a huge ethical query in itself.
I missed that in the brief. What's
It
Harriet
wasn't
Lacks?
in the brief. He's clearly been reading up, Joe. No, so Henrietta Lacks was a black African American individual who had a terrible tumor, and she died, but the tumor grew so well. The cells were growing so well in the dish that they became the mainstay cell for research to do research on, because that just grew really well because it was a cancer, right? So at the moment, tons of our cells are grown and are stored around the world, and it's one of the most used workhorse lines in research. The problem is, of course, that she never saw 1 cent of that. She never gave consent that her cells could be used for that purpose, and that's why Joe is bringing it up, because that is clearly unethical, right? So yes, she could not withdraw anymore after she donated them because she didn't have a proper informed consent form signed. But at least for the research that we do, people sign an informed consent form where they sign to indicate that they understood the conditions and also that they can withdraw at any stage.
So, if just for a bit of clarity, in the case that you take some of my umbilical cord stem cell and you run some tests on it and say, are you saying that I'm predisposed to a certain condition? Are you saying that I'm gonna have it eventually?
That depends on the mutation, right?
Right. Okay.
So there are some reportable mutations. For example, a mutation in BRCA1 is what people most know best, because Angelina Jolie, remember, she had to have her breast removed because she had the BRCA1 mutation, and she knew, like with 100% certainty, she was going to get breast cancer.
Right.
So when you have a mutation like that, it is 100% predictive. That's very rare.
Mm-hmm.
So in most cases, when you sequence the DNA, you can say you have a 12-fold increased chance of getting Alzheimer's disease, and you have a higher likelihood to get cardiovascular disease, right? But it's not saying like this is written in stone. That's very rare that your DNA says that it's written in stone. That's for most of the general population. Mm-hmm. The people that we cater most for are children that are born with genetic mutations that lead to very, very severe problems. In our case, in the central nervous system.
That's pretty close to home, isn't it? So in terms of, our family, I think in our conversation before this, I was telling you about how we've got that hereditary
I didn't
condition
sign the
of
waiver, Joe.
pheochromocytoma.
Oh, you do have the pheochromocytoma.
Yeah. Where essentially this, if you would prefer not to talk about this, I can edit it out. But essentially, with everybody outside of my sister and myself, to put it in the way that the doctor explained it to us, their genetic sentence has a spelling error in it.
Yeah.
And it's still coherent. However, there's a word in there that doesn't quite fit in, whereas my sister's and my genetic sentence is very coherent. So I think within the family we've had neuroblastoma, pheochromocytoma, ganglioroma, and then there's another one. But in that, would we, in this whole world, kind of putting everything together that we've talked thus far, in a world where you've got some sort of a hereditary condition like that, and you have kids within a family predisposed to that, and you find stem cells within the mother. Are there ways currently to nip that in the bud before, I guess, the individual grows up?
Right.
And that would have ethical. This is where it becomes interesting because there's the knowledge element and then the action on the knowledge element.
Exactly. So what if you would have been conceived via in vitro fertilization, right? There you can actually screen the different embryos for mutations like that, right? Or you can do a chorion vili, you can take some cells, when the mom is pregnant and they are then fetal cells, you can sequence those and then you can look really, really early whether you have the right spelling of the word or the wrong spelling of the word. So the sooner you find this out, the sooner you could start considering doing what's called gene therapy or genetic therapies, and that's what you're talking about. It's like, well, let's respell the word correctly, right? Let's either put the gene with the correct words back in, right, and replace the defective gene, or go in with scissors and really precisely change the wrong letters so that now the sentence is correct again, right? And that's using CRISPR or something like that, like genome editing technology. So the problem is that you need to do this really, really early in development, right? Because you want all of your cells in your body to be genetically corrected, or you deliver the corrected gene to you now, right? But now you could actually do that. You could replace, your defective gene with a correct gene. There are still risks at the moment in doing that, though, right? First of all, you can't be 100% sure that you're going to hit all the cells that are going to potentially cause a cancer. You're unlikely to do that, right? You guys are big boys. There's a lot of cells in you, right? There are trillions and trillions of cells.
There's more in Tom.
So to think that you can correct a particular genetic mutation in all the cells in your body, right, is just not going to be feasible at this stage. Maybe somewhere in the future with some whiz-bang technology, but at the moment that's very, very difficult. So having said that, CRISPR is getting better and is getting faster and more precise. And CRISPR are basically smart scissors that with a GPS capability that allow you to go into a cell, find the spot in your DNA where that word was wrong spelled, and then basically cut that bit out or replace it with the correct bit.
I cannot believe that this stuff exists.
Yeah.
I heard about that for the first time this year.
It's insane.
It's wild.
Because I really specifically want to talk about CRISPR.
Okay.
But before that, I want to preface it. You had some insane discoveries to do with brain organoids and AI.
Not AI.
Oh, no. But in terms of the Pong specifically.
Sure. So I mean, the Pong work was really started by another group and is still being done by another group in Melbourne. Mm-hmm. But what we're talking about, or what you want to ask about, is the computational ability of brain organoids, right? So you can, at the moment, what we do. Let me take a step back. So a brain organoid is about the size of a lentil. Okay, so they're not huge, but they're not. You can see them by eye. So normally, when you want to listen in or affect the activity of a human brain, you put electrodes on the outside, and you either listen in or you stick electrodes in somebody's brain and you stimulate it, Elon Musk style, and you can start to do stuff, right? So we do the opposite. We put our brain organoids straight onto the electrodes. So we have 6,000 electrodes, and then we put our brain organoids on there, and then we listen in to the neural activity of these couple of millions of neurons as they talk to each other. And then from the signals, we can figure out how active they are, who they're talking to, how hard they're talking to them, whether that talk contains useful information. So now you can start to teach an organoid certain behaviors. You can reward or you can punish an organoid. So if you want to reward an organoid, you give it patterned activity. Our brains love patterns, right? I mean, we are pattern recognition machines. That's how we evolved, because you can recognize a tiger by a whisker and a bit of tail, you're likely to survive, whereas you don't, you die.
Yeah.
So pattern recognition is what our brains do. So when we give patterned information to a brain organoid, it goes like, oh, I've been a good boy, right? But when it does the wrong thing, we give it random information, and it goes like, ooh, daddy's angry. And it knows that it's done the wrong thing. So that way you can teach it certain behaviors, right? So now you can imagine you can do that with two brain organoids, right? You can have one brain organoid sitting on one chip and another brain organoid sitting on another chip, and you can connect them via the wires, right? So you can now have an input in one brain organoid, activating a certain neuron in another brain organoid, and vice versa. And then there is us that have stimulatable electrodes. They're like, we can punish them, or we can reward them, or we can also stimulate them, with a certain pattern, and see if they can interpret that pattern as information. And long story short is that brain organoids can learn patterns really, really quickly. And even if the fact that they're only lentil size, they learn something like Pong, in 3 or 4 minutes, right? But AI, it takes so much longer. Whereas a little bit of wet, lentil-sized brain organoid from Joe can learn this in no time. So it tells you a number of things. One is that brain organoids can learn, that learning is something that's inherent to neural networks, that we can mimic that in a dish, and now we can start to learn the rules. Is how do neurons talk to each other, right? So I think that's pretty cool. And then one of the things that we can do, and what we've done is, for example, we can give anesthetics, right? Because we're really interested in consciousness, what consciousness is and where does it come from, right? Is it something that's distributed over our whole brain, or is it something that can emerge from a small clump of cells already? So one thing that we do, like, okay, well, let's make them unconscious and let's give them anesthetics. So then we give them different anesthetics and we see, they go to sleep, and we can wake them up, and all exactly at the same concentrations as that we know will put your brain under anesthesia, exactly at that concentration. Our brain organoids go like, we're asleep. And then they wake up again. So again, they behave like our brains, right? But you can imagine, like, now we can start asking questions like, okay, so if you are under half anesthesia, do you still learn the same way and as fast, right? And how does that relate to the neural pathways? But we can also ask other questions, for example, psychedelics, right? Psychedelics, magic mushrooms, people know that it allows you to learn new behaviors, right? This is how people get rid of smoking, or PTSD, for example, after you've been to Vietnam. It opens up the plasticity in the brain somehow. And it also does weird hallucinations, but it certainly does that. So that's one of the things that we are doing now. We give those brain organoids psilocybin. The first thing we see is that neural networks that were previously not active in these organoids are now becoming active immediately. So it recruits all kind of other networks in the organoid. And that's kind of maybe what psychedelics do, right? They make connections between neurons in our brain that are not normally connected. So that's the kind of games that we're at the moment playing. Like, can we get insights into what consciousness is? Can we mess with consciousness with psychedelics, something that changes perception and consciousness? How does that affect memory and learning? How does that work on a cellular level? How does that work on a molecular level? And we already know is that for example, psilocybin also increases the rate at which new brain cells are being formed from stem cells. So that's another thing that we would like to test for people with traumatic brain injury. Like, can we use that to as a new drug? And can we do that without hallucinations, for example, right? Which of the many compounds in magic mushrooms is the one that's doing the job? It's not just psilocybin. There's a whole bunch of other stuff in there that may be also doing stuff. So that's just one little flavor of how you can start to use brain organoids to get really fundamental insights into how our brains work, but at the same time use that knowledge to improve outcomes for people. Right.
And just on that, because I know you've probably got more to say, and there's way more that you want to cover, but pattern recognition and consciousness. There's an awful lot in between there. And consciousness is that is such a massive topic. But parking that, going back to just what is the organoid doing? So when it's playing Pong and you say pattern recognition and responding to different stimuli, is it literally like I guess all you're doing is correct me, and that you're watching it, you're stimulating it, and it's responding to what you're doing to it. Is that thought? Is that just like, what's going on with the thing?
Well, so I don't think it is thought, right? This is something that we can debate, actually. I mean, because I don't think we know the answer to that. I don't think it's taking a conscious decision here. I think this is a property of a highly interconnected neuronal network with positive and negative feedback loops. You know, the whole thing is sort of hanging together and sort of works as an integrated machine. I think it's just reacting to stimuli and doing the right thing to get the reward. That's certainly the lowest hanging fruit possibility. Now, will it ever be able to understand more complex information and come up with new stuff, for example, right? Because that's what consciousness does, right? It makes new connections and come up with new answers. Whether it can do that, I would be surprised if a little brain organoid can do that. But maybe if we start to connect lots and lots of them, maybe at some stage it will be able to have that ability, right? I mean, the fact that it learns so quickly, I don't know. It is not an answer to your question.
That's super interesting, yeah.
Because one, we don't really know what consciousness is, right? So what is the difference between a consciousness and...
Is it even in the brain? You know, I was reading some papers on this the other day, and it's a massive debate. Is it like helomorphism and the soul sort of being the source of, and I guess that's one school of thought. But I just find it so interesting to, like, if you look at the brain organoid in isolation, and even saying it's doing what a brain does is pretty freaking interesting.
Yeah.
Like, brains do things.
Yeah.
That's really interesting.
I know, and as I said, this is why I love that as the main focus of our lab, because it's really the final frontier. Now whether an organoid will ever have a sense of self, right, or of individuality, is a question, and the second question is, like, how would you tell, right, that it does? So bioethicists and we and the field is hotly debating this, like, at what stage and what test would you do is to stop brain organoid research, right? Because if they have a sense of individuality, or we don't think they can feel pain because there's no pain neurons connected to brain organoids. But can they be unhappy? Maybe they can be, right? I mean, maybe they are a little bit of brain that are desperately looking for sensory input, right? Maybe they are these locked-in people. Right? I don't know if you've ever heard of it. This locked-in syndrome, where people cannot communicate with the outside world, but in their brain they are just like us, but they just cannot communicate. They're locked in. Is it like that? Is a brain organoid, if it's like that, that would be super cruel, right? Because maybe they are thinking stuff, but then again, they have never learned anything either, right? They've never been born. But if we start to teach them stuff, right? Can we teach them more complex stuff at some stage, right? I don't know. Right? It takes a human, well, I guess the exact time is debatable. Some say like 4 years, when a human becomes sort of rational, right?
And it stops at 21.
Yeah, some say at 16. Some say it never happens. But But clearly our brains are very slow developers, right? So I don't know. Can you remember when you were 1 year old? Can you remember anything? I can't. So what about the brain organoid that still has a prenatal a makeup, because they're very much fetal still, right? This is the other fascinating thing. When you make a brain organoid, a 3-month-old brain organoid has the cellular makeup and the attributes of a 3-month-old fetal brain. At 6 months, they look like 6-month-old neurons, and at 9 months, they just look like newborn neurons.
Wow!
So I told you that there is this program built into the stem cells that just builds a brain.
Can they die?
Yeah, of course they can die. They die really easily.
Yeah.
Yeah, just like our brains.
Yep.
What's the life expectancy of a brain organoid?
Well, we have ones growing for 3 years now, but other people have grown them for up to 5 already.
And do they? And sorry, when I say do they die? Let's say you have it in the ideal conditions. The life expectancy. That's probably a good way of putting it. It'll naturally die?
No. Nobody's grown them for 70 years yet, right? Because this technology is real new. But maybe they will. We do know that they age, right? This is one of the things that we've done. Is one of the cool outcomes of our research last year is that we infected brain organoids with COVID-19, right? Because this happens, right? And this happened, and people have brain fog and have long-term bad side effects of COVID-19. So one of the things that we found is that it induces a process called senescence. Which is basically tells you that certain cells in your brain are becoming much, much older, about 10 years older than they're supposed to be after a viral infection. So a viral infection makes your brain older. And then we showed that particular compounds called senolytics that kill these senescent cells, can help and can improve brain health and brain outcome. So there's a number of lessons there again, right? You can play games with brain organoids that you couldn't play with humans. You can't deliberately infect humans and their brains with viruses to find out what happens. We can. Secondly, you can't screen drugs on those individuals to find out, something that would help against a COVID-19. And at the same time, it tells you that certain aspects of aging are recapitulated in this model as well. So why did we get to aging? Oh, yes, do they die? You said, do they die? Yeah, eventually, so we don't know. Eventually, they may die just of old age. But remember that they are not connected to an immune system or anything else, right? So maybe they will just keep going. I don't know. But at the moment, the old ones are fine and they're happy.
So 3 years at the moment is the oldest one that you have that's alive right now. So you're not actually sure of the life expectancy?
No, I don't. I personally think that it will depend more on how much money we have to keep buying media and growth factors to keep them alive, rather than anything else. And I'm not sure that we don't do this because we have a question like that. We do it because we are interested in whether they are getting more attributes of a 3-year-old brain than a fetal brain. So and because that maturation of the brain, is something that is really important in humans, right? And again, we know very little about that. So how did we get there? Age, organoids, lifespan. I don't know. And now I've lost my train of thought.
Before we deviate, Christmas to come. You were alluding to the use of psychedelics earlier. Have you used or tested psychedelics on brain organoids, and have you found out some pretty crazy but also useful stuff? Because I know that psychedelics as a source of medication, particularly in a military aspect where you're treating soldiers with PTSD with psychedelics, is becoming more normalized. Is there stuff that you're finding out about psychedelics that may change the future in how, I guess, we look at psychedelics as a drug?
I certainly hope so, yes.
Whoa.
Because as I said, at the moment we are already treating those organoids with psychedelics, and one of the things we see is that if you have an active neural network, and when 2 seconds after we give the psychedelics, other neurons that previously were not talking to those neural networks are now talking to those neural networks for some reason.
They want some.
So they recruited, they recruited into the network, right? And go like, come and talk to us for some reason. We don't know how they do that, but it certainly happens. So increased neurogenesis, I told you that, it produces more brain cells. And we're starting to look at the impact on learning and memory in brain organoids. I think that there was just a paper coming out last week that I had found another compound in a different type of magic mushroom, but also did that. It enhanced this ability to learn, or to this plasticity, as they call it. Being plastic, being open to new learning without being hallucinogenic. So clearly that's happening now, and there are several small companies that we work with, and the AIBN works with, where I work here in Queensland, that are producing psychedelics and psilocybin. And it's a burgeoning industry, and Australia is one of the few countries that has just allowed, at least under very strict rules and regulations, for psychologists to prescribe psychedelics for certain cases. So we're kind of cool, Australia there, right? We're kind of at the forefront and allowing stuff to happen, but normally we're very, very conservative, and everybody was very surprised about that. But maybe there are certainly opportunities. I think there's going to be massive opportunities actually with psychedelics and other compounds like that, to find out what else they can do except for making you see. What was it? Mechanical elves or something like that.
Mechanical elves?
I was at a tech conference.
That's what Joe Rogan said.
Yeah. But I was at a tech conference recently where there was a gentleman from Melbourne who has basically a psychedelics therapy / VR virtual reality treatment center. That's definitely downplaying what he does, but basically the idea behind it being somebody goes on a psychedelic experience and they're all wired up while they're doing it. And there's all this AI mapping the neural activity while they're undergoing the psychedelic experience. And then after that, the idea is that the patient, through virtual reality, re-enters the artificially recreated psychedelic experience. I don't know how accurately it would map, but in theory, they should be able to then kind of, as an external, as themselves, re-enter what their brain was going through and identify things. And I evidently I've never done it. But it's just, it's kind of...
I'm not sure they're going to work. I mean,
I don't know.
From what I've read, psychedelics is a little bit like when you're dreaming, right? So when you try to remember your dream, you can't remember it, right?
Well, they just make no sense. Mine are bizarre. I have weird dreams.
No, but have you noticed that when you wake up, when you're sort of half asleep, you can sort of still remember, but the more you try to remember, the less you remember, right? So I understand psychedelics are a little bit like that. So I'd be surprised if that works, but maybe it does, right? Maybe that's enough to trigger some memory pathways. But I think I do know which institute that is. They do some cool work. But so we are coming from a very much different angle there, right? We're coming from a very fundamental level where we want to figure out. Here we go again, right? Psilocybin been around for like, how long have we taken this? 10,000 years, right? So some people think that this is the reason why we walked out of Africa, right? You do know about the stoned ape hypothesis, right?
Talk to me.
Oh,
yeah.
I mean, one hypothesis or one idea is that as we came down from the trees in Africa and we came onto the savannah and there were lots of large animals dropping their poo there, this is where where these mushrooms are growing on. Somebody went like, let's eat those things as food. They had a hallucination, and they went like, oh, a stick. I wonder if I can use that for, right? Because it makes neural connections, new pathways. So this is one hypothesis, is that one of the reasons why we were so successful, and it stimulates brain growth, right? Because this is also when our brains all of a sudden start to grow massively during the time that we just happened to be on a savanna with magic mushrooms. So this is all hypothesis, right? But it's a kind of a nice idea to think that maybe magic mushrooms were both driving the growth of our brains and the idea of doing stuff that we not normally would have considered doing.
Yeah.
And now, what is it, 30, 40,000 years later, or maybe 100,000 years later, we are rediscovering, that these magic mushrooms can do really cool things, right? So yeah, I think that's good. I think there's a lot more to be learned. A lot more to be learned.
And it's a promising future for psychedelics.
Coming from two guys that have like bucket loads of experience in psychedelic use.
I didn't.
You guys are so strange. For an Amsterdam boy, I know you guys are so straight.
Our backwards caps are as cool as we get.
I'd put mine on first. So CRISPR.
Yeah.
How long have we been going for? This is opening a can of worms. You don't have any place to be right now?
No, then I don't have to cook.
Don't have to cook? Sweet. CRISPR. What's CRISPR?
So CRISPR was discovered not too long ago, but it was discovered as the immune system of bacteria. So bacteria, just like us, get infected by viruses, right? And like everything gets infected by viruses. But bacteria don't have an immune system like we have, obviously. There's only one small cell. So how does a bacterium deal with an invading virus? Well, what it does, it takes the RNA or the DNA of the virus and it chops it up into little bits. And then it basically uses that information to design a tool to destroy anything that looks like it, right? So let's say you have a virus that has the sequence, I'm going to make this really simple, ABCDE, right? Now, this virus enters a bacterium. The bacterium cuts it up into little bits, and there's lots of ABCDE little bits in there, right? It now remembers that bit, and it mounts it into a tool that says, like, anything that looks like this, we're going to cut to bits, right? So now a new virus comes in that looks like that. The bacterium is already armed with this particular CRISPR, and that's what CRISPR is. And it goes like, oh, this looks like that virus. Let's just destroy it. It's not ours. It comes from the outside. It's a virus. We need to kill this, right? That's how it was discovered. So basically what it does, and so that's the GPS smart bit of the system, right? That it can recognize other DNA sequences based on homology, on how similar they are, right? If it is ABCDE, it goes like, oh, ABCDE, that's the same as what I want to destroy, what I want to cut. So it then binds to that and cuts there, right? So that's how CRISPR was discovered in bacteria. Then it was really the smart thing to do, though, was to like, okay, we can hack this system and use it in our cells. So now what you can do is we know the whole DNA sequence of our DNA, right? And I can sequence your DNA in a day, right? Now that used to take 10 years, now a day, and $800. So now I know your entire sequence of your DNA. Now I can say like, okay, this little bit here, I want to start cutting that because I want to insert something there, or I want to change something there, or I want to add something there, right? You need to find that bit. So now I say like, okay, I'll take that little bit that looks exactly like that. I put it in my CRISPR tool, and it will now start to hunt all your DNA and go like, there is the bit that looks exactly like what Ernst was saying I was supposed to be looking for, right? Yeah, just like what happens in the bacteria. Now it's not a virus that it's looking for, but it's looking for a bit of your DNA. Now it binds there, and it goes like, okay, this is where we cut. It thinks that it's a virus, but it's not a virus. It's your DNA, but it'll still cut there, right? So now there's a break there in the DNA. Now you can do a number of things. You can leave it like that, and that means that there's going to be damage on this particular site. So wherever that cut, that gene is no longer going to work, right? So you can use that to knock out, for example, a cancer gene, for example, right? If you can deliver that to the cells that will give you your glioblastoma, right? Then you can, then you could specifically stop them dividing, for example, or kill them, because you know exactly which bits of DNA you need to attack, right? Or you could have a mutation that gives you some disease, right? It's like sickle cell anemia, for example, right? Or beta thalassemia, it's a blood disease. You don't carry oxygen well. There's lots of people in the Middle East have this. It's in your blood, and your hemoglobin doesn't work properly. It can't carry oxygen properly. So you want to fix it. So you come in with the CRISPR, you look for the hemoglobin gene, you find the bit where the mutation is that prevents it from carrying oxygen, and you go like, okay, we'll replace this bit by the right bit. Now you've fixed the hemoglobin, now you've fixed the beta thalassemia, and you're good. So that's how CRISPR works.
Is it from a biological perspective unusual that something like that is or was unique to bacteria? Because that's so... No, I'm not even going to say bizarre. That's just so interesting to hear that it was from bacteria.
Bacteria have been around for such a long time, right? I mean, like 3-4 billion years. They had a lot of time, and they've been always living together with viruses and things that want to invade them and kill them. So I think bacteria was kind of a logical place for a system like that to evolve. But what's surprising is that it's hackable, right? Huh. That it works in our system. So now you can do all kind of cool things with CRISPR, right? Because it's got this GPS capability of finding the right bit in your DNA. You can use it to visualize where this bit is in your cells. And go like, okay, where in the nucleus is it? You can say like, okay, no, I want to cut it and destroy it. That's easy. That's how you knock out a gene. And my students do that all the time. You might want to repair it. So like, I want to change this A to a T. Right, so now you can fuse your CRISPR. Yeah, you say like, don't cut anymore. I don't want you to cut anymore. I want you to replace.
What?
So you bind it to an enzyme. An enzyme is nothing less than a protein that does a job. A protein that is a replacer. It's a DNA replacer. So now the CRISPR says, like, this is where you need to go. I'm your GPS capability, and the enzyme you bound to me allows me to really specifically change this A to a T without cutting anything. So it can do that. You can also change the way that DNA works is that it's a linear molecule, but it's a linear molecule, but it's totally wound up, right? It's totally packaged, right? So you can also use CRISPR to unpackage particular pieces of DNA at will. So CRISPR is like a multifunctional Swiss Army knife for genome manipulation. You can edit, replace, move, visualize, mess. You can mess with the DNA really easily, really quickly, and that has changed the game. It used to be very difficult.
So that's what they used for the direwolves.
Yep.
So on I guess...
We're back at direwolves.
No, well, yeah, we are back at direwolves. It's
all connected.
It's all connected.
It's all
it's insane. I guess, and on what's the word? Like in the terms of treatment on humans at the moment, to what capacity are we using CRISPR?
So not much yet, because what we talked about is that it all depends on how efficiently you can deliver it to the cells in your body, right? So where it's used at the moment is, for example, for beta thalassemia, because what you can do is you can all the bloods that you make in your body, you make about 2 million blood cells per second, in your. Yeah, I know, it's a lot. So what you can do is they live in your bone marrow, right? Those blood producing cells. So you can take the bone marrow out from somebody. You can destroy the rest of the bone marrow so it doesn't make the bad blood anymore. Now you can take those they are stem cells that make your blood, of course, right? You can take those hematopoietic stem cells or blood stem cells, grow them in a dish, and then use CRISPR to correct the beta thalassemia mutation, right, or the hemoglobin mutation. Now you select the one that has the right correction, right? That has the word corrected properly. You can just test that at your leisure. Now you grow up a lot of them, and now you put them back into the patient. And because they're the only blood producing stem cells that the patient have, they will lodge again in the bone marrow, and then make blood that doesn't have the disease anymore. That's one way you can do it. So that's an ex vivo CRISPR, right? So you take the cells out, you mess with their genome, and then you put the cells back. What the ultimate is to do in vivo CRISPR, where you correct the mutation in vivo, right? And that's only being done at the moment for, I think, retinal pigment epithelial cells. So these are the cells on the back of your eye, which make you blind, which gives you macular degeneration. I don't know, some you may have heard of macular degeneration is with the macula, the back of your eye degenerates. You get it when you're old, and in some people that is caused by a particular mutation. So because you only have 1,000 or 10,000 cells on the back of your eye that do this job, that was the low-hanging fruit where they tried in vivo CRISPR, because they injected the CRISPR in the eye, they then delivered it only to those 1,000 cells, they corrected the mutation, and then it worked. So they tested it out in mice, and then in rats, and then in monkeys, and now again the first clinical trials are happening with that. So that's, to me, to my knowledge, the only in vivo CRISPR work that's been happening at the moment.
Also, clinical trials are underway.
Yep.
Because there was the, I guess, in China with the HIV, was it HIV
You
babies?
have done your homework, mate. Yes. So that was a different situation. That was actually more closely related to the cloning issue that you talked about, right? So there they did use CRISPR. And what he did is he basically genetically modified the kids before they were born, right?
Yeah.
So this was all done, before they were implanted, right? So yeah, that was however the first two kids that were born with a CRISPR genetic deliberate mutation that supposedly would prevent them from getting HIV. Correct. That was a huge shitstorm, of course, right? Because one, that is illegal under all the rules in all the Western countries, and the International Society for Stem Cell Research is all illegal. The guy has also disappeared now.
Just gone.
Yeah, he's gone. But the kids apparently are doing okay, right? And it was only one mutation, right? There's only one base pair of the billions of base pairs that we have, right? So it was not like a massive edit of the DNA. Nevertheless, it's super dangerous for a number of reasons, right? So one is that most likely they are resistant to HIV now, but they're most likely more susceptible to other viruses, right? So that was the whole idea. Oh, we're going to make superhumans that are resistant to HIV. Well, nature, like Jurassic Park, right? This is where Jeff Goldblum was always right. Nature will find a way. So you can't, if you don't understand the entire system, to just introduce those mutations because it makes you resistant to HIV, without knowing that it also makes you more susceptible to other viruses, what we now know, that tells you that you shouldn't be doing genome editing before you really know what you're doing, right? So for something like beta thalassemia, where we understand exactly why the disease is caused, I can see this, right? But editing receptors or messing with the DNA of kids that are perfectly healthy, right? That was the other issue. They were not sick, right? They were fine. They were not even born yet, but they genetically edited them. That is a dangerous slippery slope that we should not be going on, right? I mean, we should not throw the baby out with the bathwater so that we can correct mutations like for pheochromocytoma, for example, right? And we should be able to fix those things. But at the same time, you can immediately see where the human race likes to go. Like, oh, let's put in mutations that will make us more intelligent. Right? Let's put in mutations that will make us react faster, that will make us more resistant to cold, that will give us longer endurance, that will give us more muscle mass, that will make us look prettier, that will give us blue eyes. We've been on that road, right? 60 years ago. That was not the way we wanted to go, I thought. But human vanity and human desire, optimization of your offspring, right, is kind of a human thing to do, right? So there's lots of push of people, and rumor goes like in countries like China, for example, there could be people that are now deliberately asking for these edits, like I want edits for being musical and for being sociable.
And then you have a supply and demand problem, and then you have. Yeah. Money.
So that's one issue. You get a society of inequality, of haves and have-nots that can and cannot afford it. You're creating a super class, like Gattaca style. You are not using it for what it's supposed to be used for, right? It's for curing diseases, but you're using it to improve a very personal, select a group of individuals. That's one issue that is clearly a problematic. The other one is that, for example, you guys have heard about this whole autism thing at the moment in the States, right? Like, what was it?
Tylenol.
Tylenol. You know, pregnant women can't take Tylenol, and I think the evidence is very bad. But there could be a small effect size, but whatever. It's very, very, very small. But people always say like, oh yeah, no, we need to use CRISPR to prevent people from getting autism, for example. And certain mutations, certainly cause autism more, and we know there is a causal relationship. So now let's say that you have a baby that's being born with its mutation, right? So do you want to either not have it born because it has the mutation, or do you want to fix the mutation so that it doesn't get autism? And many parents might go like, yes, that's what I want, right? But Einstein also had autism, right? I mean, this is the most beautiful example of a person with autism that you probably don't want to edit your genome, right? And maybe the question is, would he have had the same insights and thoughts if he wouldn't have been autistic, right? I think it's very likely he wouldn't have.
He wouldn't have been Einstein as we understand
exactly.
it. Yeah.
Exactly. So curing, and this was clearly an issue also for the parents, right? In the United States. They're like, well, there's nothing wrong with autistic kids, right? But why do we have to be so scared about Tylenol? There's nothing wrong with autistic kids. Now, autism is not a funny thing to have at all, but it highlights that this diversity that we talked about in the cows, right? This diversity is also very important for humans, right?
Nature's way. Yeah.
If you in 200 years, if genome editing has become so prevalent and so common procedure, like going to the dentist, then eventually all of our DNA genomes are going to be looking similar, right? Because everybody will want to be slim and tall and long legs and smart and good at everything, right? So all of our DNA is going to look very similar, right? So which is an invitation for viruses to wipe us out, but also for our societies to become very gray and very similar to each other, right? I think this is one of the good things about human species, that we are so diverse. This makes us strong and interesting, right? This is where we get diverse insights and opinions. I think there's a real risk that we're going to make everything perfect, right? Like everybody looks like Barbies and Kens, right? But that's not. To me, that is not a strength. That is a weakness, right? So that's something that I think we should not forget in this whole CRISPR editing debate. Is that if you have a mutation that will give you cancer or a disease, definitely, right? But we should have some serious regulations about people who want to edit their babies for desirable traits that society somehow has decided like are either no good or a skin color that we don't or an eye color that is non-desirable. Right? I mean that's it. This is where we really need to be vocal, right? And the scientists are vocal, but the science is running ahead, like at breakneck speed. The politicians have no idea what is going on. The society sort of reads something in the paper maybe or online. Sometimes we go like, oh, I didn't know you could do that, right? I go like, yeah. I mean, we need to start thinking about that now, right? We need to start thinking and educating people and start thinking about how we're going to deal with this. So that's one of the reasons why these podcasts are important, because people need to start thinking about it and talking about it over the dinner table and go like, what do you think, right? And what would you and on the, because it's a very slippery scale, right? I mean, there are also conditions where you know that you're going to have more chance of getting a disease, but on the other hand, it's also possible that it will improve your connectivity in your brain, right? So do you correct that one? Huh? Someone who go like, yeah, I don't want to have liver cancer. We go like, yeah, but you're also going to get less smart, dude. So like, well, if you can hit my liver specifically, fine, but maybe not my whole body then, right? So you can see that there is always this like, okay, do I want, is this all right? Is this possible? What do we think as a society about that?
Sorry, have you?
I'm done.
You may have addressed this, but in theory, what it does functionally makes an awful lot of sense, but that's also ridiculously invasive. Are there side effects of going through that
CRISPR?
process? Yeah, and I think you were sort of just unpacking that, and that's sort of what's going through my head. But can you just sort of focus on that element?
Yes, I mean this is one of the reasons why it's not in mainstream use yet in the clinic is that CRISPR is pretty good at recognizing where it should cut or bind, but it does. There are other sites that kind of look the same, right? They look 90% the same. So CRISPR will sit on that for some time as well. The enzyme will sit on that and go like, yeah, almost right. But in some cases, it will cut there as well, which they call off-target activity, right? So undesired off-target activity. So this is a big issue, and we know this happens. And we're actually designing, one of my PhD students has just designed a new tool where we try to mitigate against that. But this is a known problem with CRISPR, is that you do get mutations elsewhere in your DNA while you're also correcting the thing that you did want to correct. So yes, eventually this will become more precise. Undoubtedly, it will become more efficient. But at this stage, this is one of the bottlenecks, and maybe it can never be made 100% specific. So you will always have to live with some damage while you're doing your desirable edits. So yeah, that's sort of where it's at.
Yeah, and I think this is one of the crazy things, to use beauty as an example, is beauty standards as a result of social media. And since social media has, I guess, blown out of proportion what is normal attractiveness, a level of attractiveness, all to do with, like making it seem that the whole world is, or everybody in the world looks a certain level of attractive, has also decreased what was once attractive and moved that benchmark up, but also created a benchmark where 90% of the world is falling short of that. So you're looking at a super normal person or a supermodel for someone to even trigger any sort of mental damn, she's attractive.
Or he.
Or he. Or he. But it's interesting. I guess the point that I'm trying to make is, prior to something like social media, for example, where you weren't exposed to individuals at such a large scale and diverse scale, your friendly neighborhood girl or guy was a lot more was just a lot more attractive to you. You didn't have unreasonable beauty expectations.
No.
And you weren't projecting those on yourself or everybody else around you. Yeah. And then when a girl sees, and there was a paper done on this. This is where I was trying to go. With beauty expectations and standards, girls, and specifically with girls, but definitely with guys, but they try to imitate the what is culturally attractive and fashionable at the time. And, we've been doing that for centuries with clothing and all that sort of stuff. But now in terms of, where we're getting into like facial reconstruction and the science, but the medical and the capacity for that sort of change, accommodating that has pushed people. Well, I've been dribbling on. Hold on, I've lost my spot.
I'm totally locked in on this.
I've lost my spot. Has made people more of the same, and in pursuit of this thing that they thought was going to make them more attractive, has actually just brought the bar down. Does that make sense?
I think so. I think so. But yeah, it goes to this point that everybody's starting to look the same because they all have the same beauty standards, right? And that everybody's and yeah, I mean, I was talking about that with my partner the other day when she said like, I need to dye my hair again. I go like, no, you don't, right? And there's nothing wrong with gray hair, right? And that's another one of those beauty standards, like, oh, you can't have gray hair. I go like, well, in the old days, everybody had gray hair,
right? Yeah.
And I think it's beautiful, right? And there's nothing wrong with gray hair. It's just, it's why people want to do something about that, or why aging is something that you're not allowed to do anymore either, right? You now have to stay young, you have to look young, right? All of that comes from social media or from society. It's just that information flows so much faster now than it used to. But it does raise a question. Something that I was just thinking about that we should talk about as well is like, as you know, it's 50 or 60% of what's online is all AI generated, right? So it's AI bots reacting to AI bots, and humans sort of looking over the shoulder and go like, oh, well, I should look like that, even though it's probably not even real, right? What they see is not even real. And that's something that we should also talk about, because that is what is changing biosciences in a big way at the moment is AI. Like big data, right? AI can make sense out of big data, pattern recognition in big data. Designing novel drugs from scratch is now done by AI. The AI can read all the literature on a disease and make predictions, that we don't know how it came up with those predictions, but in the few instances where it has done that, the hit rate was pretty damn high. So AI certainly has something to bring to science as well, But on the other hand, I think most of the grants are now written by AI and are being reviewed by AI. Articles are being written by AI, right? Scientific articles. In science, just like in society, it's becoming increasingly difficult to tell what is real, right, and what is not real, right? And if in science if that happens in science, now we have a problem, right? Because we deal in facts, only in facts. And I can see this is where both society and science is now really struggling with we like AI because it can help us, but on the other hand, AI is sort of inhibiting us. If you see what I mean?
In terms of our own like individual learning and progress, or inhibiting?
No. So AI can look at big data and pattern recognize. And that's why we like it. But at the same time, AI is making it very difficult for scientists to determine what is real data and what is AI generated data.
Right.
And the same thing is happening in society now, right? On the internet, as we just said, the half of it is just BS. It's not made by humans, right? And it's not information that is parsed by humans. So, that in a time when science itself in society is looked upon with some frowns, right? Who is trusting scientists these days, right? The conspiracy theories abound about scientists, like they're in the pocket. Fauci is this, These people, I know Fauci. They are scientists at heart. They go by the data, right? They don't make stuff up, right? That's what scientists don't do. But now they are being challenged. They're like, oh no, you don't know what you're talking about, right? Because I read online that, and my AI engine says that, and yeah, I can see that as a problem in the future going forward, when both society and the scientists themselves don't know what's real anymore. I think we have to think just like CRISPR. AI has its uses, but we have to be very careful, right? In what we're using it for and what we are relying on, and for what we are relying upon, what we are relying on, in terms of technology. I think this is. I can see this as a big problem going forward.
And you wonder if there will be just a natural correction over time, because I think if you look at, take for example, marketing, and you had the digital revolution, the AI revolution, and then I recently read a paper where I think it was for at least the first couple of financial quarters for 2025, the number one determining metric for a campaign success was, did the campaign generate trust? And there's 50 million different ways you can look at that. You can look at, user experience. You can look at the actual belief the consumer had in the campaign. But once you start working in trust, you're really you can utilize AI, but you're going back to logistics and sources of how things are put together from the ground up, and it all becomes an awful lot more three-dimensional and ethical. And AI is really, really fascinating and cool, but is it at that stage where it's doing anything more than just really fast-paced deductive reasoning? Well, I guess you do have agentic AI ecosystems, which is kind of freaky, where you've got basically companies that are run by AI agents.
Our company is.
That's freaky. That's freaky.
Yeah, I know. Saves cost though. It does it much faster.
Yeah.
I mean, all the robots and the software that we design that we use for the robots in our company is all AI generated. I mean, and there's no competition for it anymore, right? We were the CEO and I were sitting on the tram in Rotterdam from the train station to the building, and in 6 minutes we had code design and test a new software routine for our robot, and we load it and it works. In the old days, you had to have a software company designing this and testing it, and now this is done in 6 minutes while you're riding a tram in Rotterdam. So that is a game changer, and the fact that it can do data analysis, it can do data integration, it can do all the marketing, comms, finances. I mean, yeah, I think this is where AI is coming into its own, right? It's where it starts to provide the information that billions of people rely on, right? On X and on whatever social platform you want to do, that's when I think it becomes problematic, right? Let alone when it becomes potentially conscious or, you know.
I wonder though, I don't know how, given the rapid developments of AI, and you talk about, you know, the what do they call it, the singularity and all that sort of stuff, that, well, whatever. I mean, it could be, but I imagine we're not quite there yet. But at least a short-term safeguard is the question of liability. So long as those behind the AI are still held accountable, whether or not there are questions of whether or not you can pierce the corporate veil to get to
I don't think it's been
those
tested
people.
yet.
I don't think it's been tested, but gee, it's a freaky world, isn't it? On all these topics, because
Spoken like a true lawyer, by the way.
So
money talks.
Yeah.
And that's the commercial consideration. I mean, you talk about all the opportunity here, but you just know that there's a lot of people with money that will use the technology to make money, which is fine in and of itself until the means that you make the money become questionable. But they're fine because they make your money.
Uncomfortable.
Uncomfortable.
Yes. Yeah.
Interesting.
So when you're talking about with AI and then scientists or in a science realm not being able to interpret what's real and what's not, and that becoming a very real problem, is that just I'm on the right track?
You can paraphrase it like that, yes.
Yeah. So the thing that makes science is theorizing and then applying it and putting it into practice and then seeing how the field work plays out, and then if it works, then pushing a little more forward, or if it doesn't, going back to the drawing board. Are you more or less talking about the trust that, you know, the society that the scientists operate in have in the scientists, or are you talking within a realm of like science and being able to push it forward?
Well, I think there are two different things, right? So for the scientists who are using AI, at the same time we are scared or cognizant of the drawbacks of AI, given that we know that it can come to conclusions and provide data that are not necessarily, you know, true or hard facts. There is that. There is the use of AI in the science more generally, where it's being used to judge other scientists, their funding applications, the review of the papers. All of that is getting done more by AI, right? And I know many of the scientists are writing their scientific papers using AI. So I find that goes at the heart of the scientific process, right? Where it's supposed to be scientists that come up with new ideas, test, design, the test and design cycle, and then publish the data. They publish the data, they make the data. And then another human looks at that and go like, yeah, I found that too, does the test and go like, yeah, we find the same thing. It shouldn't be some AI bot that reads the 500 papers that have ever been written on the subject and go like, yeah, that kind of fits with what, you know, the general consensus is, what it determines, and go like, yeah, it's all good and it should be accepted, right? Whereas there's no human in the loop anymore. That I find, and this is starting to happen now. So that takes the whole scientist out of the loop here, right? And I find that touches the scientific principle of how science should be done. And then there's this other side that you were talking about, which is the AI. It is how scientists are being viewed in society, right? I mean, that is very much a marketing exercise that we are very bad at, scientists, right? Scientists are not very good at explaining what they do or how it is relevant to society. There is that, and there are these, and I think there's a concerted effort especially in the United States at the moment, to suggest that scientists are on the take or have ulterior motives or do it all for financial gains or some other reasons, some nefarious reasons, why scientists espouse certain opinions. From what I've seen, 99.99% of the scientists that I ever encountered, they are only interested in one thing, and that's the truth. Right? They are not interested in spin or in anything else. They'll just pursue the truth. And I think that's not being recognized by society anymore. And I find that troubling. I find that very troubling that society, who are we going to trust? Right? Who are you going to trust eventually? Lawyers, probably, right? Judges. If we can't trust the judges anymore, we're in trouble, right? If you can't trust scientists anymore and their advice, we're in trouble. Because who else is going to tell you that it's, what's going on? It's not going to be the politicians. So yeah, I think this is a really important part of science that we're losing in our society, right? The respect, or not a respect is not a good word. It's more the value that people the value that people assigned to what a scientist says is eroding really, really, really quickly, right? I mean, you can see it with climate change, right? How long have scientists been saying that climate change is an issue, right? And we still have an American president that says, like, no, it doesn't exist, right? So, I mean, what can you do? How much can scientists say that this is an issue, right? But the whole narrative, right? Like, oh no, it's all made up, and it's a big cabal, and it's all for green energy. I mean, no, this is just not how it is, right? This is just hard facts, but people are throwing the facts out, and I think this is starting to happen. I can starting to see this happen now in my discipline, right? I mean, it's certain disciplines are suffering more from that than others. But yeah, I find this troubling.
Ernst Wolvetang, thank you so much for joining us. There's so much more to cover.
We have.
Yeah, we've covered so much, but there's so much more to cover that we'll definitely tackle in future podcasts. To finish off, I want to play a little game. Okay. I haven't. It
should be good.
This is called Possible or Impossible.
Okay.
So,
let's do it.
Jurassic Park. Possible, impossible, or plausible? Like you can see it happening. So, Jurassic Park.
Plausible.
Anyway,
it's possible. It's possible if you can find DNA that's intact. Yes.
Wow.
Yeah.
Planet of the Apes.
We haven't touched on chimeras.
No,
let's not do that. Let's leave that.
Chimeras are coming. There's get keen.
Possible.
Holy. Captain America.
No.
No.
I don't think. You mean what does he do again?
He's just super.
He's Captain America.
He's Captain America. He's
right.
Fast, strong, jacked,
unkillable, unblower-upable. He's just a.
Yeah, no, I don't think you can become unblower-upable, but
I think he's unblower-upable.
But I think you can certainly become stronger. Right? I mean, this is one of the issues that they are now looking at for the new Olympics here in Brisbane. Right? Is finding out if athletes have undergone CRISPR to increase muscle mass, for example.
Oh, don't say that, because
doping. Yeah.
I can't. We can't say that and not address that whole conversation.
Let's do it next time. Be disciplined.
Yeah,
you're the host.
You're coming back. You're coming back.
Make notes.
So enhanced sensors.
Yeah, I think it's in the future will be possible.
Holy cow! What about regeneration? So rapid healing.
Definitely. Well, rapid.
Or just just possible. Regeneration.
Oh yeah.
Can I throw a
100%.
regenerative enhanced brain functioning?
Yeah.
Cool. We'll talk later.
I think that's certainly possible.
So intelligence and cognition. Obviously, we talked about how you can enhance your intelligence and cognition, but I mean, to the degree that someone like has photographic memory, the IQ of, can you tap into the brain and essentially unlock even more of its power than you already have?
I would not be surprised. No, I think it's plausible.
You wouldn't be surprised. No. I would be.
Because we started to learn the rules of of neuronal connectivity. So once we understand the rules, we usually can hack the rules. So you can enhance certain neural pathways in the future in a very specific way. And if we can figure out how they're linked to intelligence, I think that's going to be possible.
Bone and changing the actual compound of and the nature of the bone. Can you?
You mean Hulk style?
Well, Hulk style, kind of like Wolverine style. Nah. Okay.
Can you unpack that one? Because I don't know Wolverine enough to understand that question.
No, so in I'm not the biggest Marvel guy, but later on they tamper with his bone composition and make it more metal.
Yeah, I don't think that's going to happen very soon now. The thing is the speed, right? I mean, I can imagine you could increase bone density and bone growth and regenerative ability, but it will never happen. Like Wolverine, you stab him and he's back, right? And that's just not going to happen. That's not biology. But for example, axolotls, right, and newts, they regrow their limbs, right? You can cut off an arm of an axolotl and it'll regrow, right? And we have lost that ability. But when you're an embryo, you actually do that. So in an embryo, you can cut something off like that and it'll regrow. But shortly after birth, about a week after birth, we lose this ability. We don't know why, but I would love to find out. Because as soon as we know why, then we can hack it again, right? So if we can bring back the regenerative ability of an axolotl in a human, right? Then you can regrow a limb again, right? Not in 2 seconds like Wolverine would, but over a year or over 2 years, right? You could regrow it. So that is definitely going to be possible, but not in the timescales of Wolverine, I'm afraid.
There are two more.
Go for it, man.
So there's adaptive camouflage, like cuttlefish.
Oh, yeah.
What?
Oh, that's coming.
What?
Oh, yeah. Oh, I wouldn't be. Oh, yes. I would be surprised if it's not already there.
In our skin.
Yeah. I mean, that would be very easy, no?
Don't know.
No, I don't know.
Tell me.
Well, if you think about engineering reflective capabilities, for example, right, of skin or of melatonin, for example, of skin, I think. Well, if an octopus can do it, right? Octopuses do that all the time, right? They can change the structure and the color of their skin all the time, right? That's how they talk. So if an octopus can do that, and we can start to understand how it does that, then onboarding those genetic circuits in a human cell or in human skin cells in the future, I can see that would happen. No, there's the tools that we have at the moment are insane, and this allows me to sort of draw this line into the future. And this is not going to happen in next year or in 5 years, what you're talking about, right? But I can see where this is going and how quickly it is evolving. As soon as we know the rules, humans will hack it. So, and octopuses. That's another podcast. That they are so cool. Hey, they.
Oh yeah. I've got epic underwater footage when I've gone diving of an octopus, and I've seen it in the distance, and I've swum after it, and as soon as I got within 2 meters, it just disappeared into its background. Yeah. It didn't move; it just became the background, and they're amazing, incredible.
But they have 50,000 genes, right? We have 25,000 genes. Right? They have double the amount of genes that we have. So
Well,
they
that's because they got 8 legs. They need more genes.
oh, they have.
Shut up.
Stop. Apparently they have brains in each of the legs too, right? I mean, and many of the genes. This is the weird thing. Many of the genes in octopuses don't look like the genes that we know about in other species, right? So you can sort of see how species are related by looking how similar their genes look like, right? But octopuses are sort of sitting out by themselves, and many of the genes go like, well, that doesn't look like anything we recognize. They could well be aliens, right? I mean, they are just so different. But that's what I'm saying, right? Camouflage. If octopuses can do it, and we understand it, we can hack it, and we can onboard it.
That's crazy.
And then the last one is flight. So wings.
Oh.
Let's go wings first.
So there are two answers to that. So I have a friend who still wants wings, and
Is it a human?
it is a human.
So you don't talk to birds.
No,
Flightless birds.
but the thing is, we still have the genes for feathers, right? So we still have the same genes for feathers as chickens have. We just don't use them anymore, right? So we just permanently shut them down. So it is not beyond the realm of imagination that we could reawaken the genetic program for making feathers and make feathers. But that's not wings. I think if we want to make wings, then we need to change our entire skeletal makeup, hollow bones, light enough for flight. Yeah, that's going to be a bit off, I think, because we don't have the musculature and to carry wings. So I think that'll be a while off, but be nice.
For sure.
I like gills. I think gills is something we should really start thinking about. Yeah, because we also still have the genes for gills, right? I mean, I know a little bit. Maybe that you guys think differently, but during evolution, right, everything that we evolved from, right, those remnants are still sitting in our DNA, and it's just that they've either mutated into something else that now don't make gills, but they make ears, or we just go like, yeah, no, we don't need the gills genes anymore. Let's just shut them down. But genes for gills would be cool, right? I mean, if we would have those, we could live underwater, we could go surfing, we could swim underwater, we could live underwater if we wanted, right?
Yeah, for sure.
So yeah, I think this is one of the things that we could touch upon in one of the future podcasts. Is that this is what inspires biohackers, right? They really like this biohacking idea and changing their bodies at a genetic level. I think as we will go further, and and if you look at the human condition, right? So at the moment I see people with metal spikes coming out of their forehead, right? And things through their cheeks and whatever. This is what people do now, right? And even, there's no utility to it either, but this kind of looks cool, right? So if you know the human condition, I go like, if we can do this, right? If somebody can make feathers, people will want them, right?
Yeah.
If people tattoos, instead of having a tattoo, you can just have your own skin cell making colors, right?
And you can turn it off when you go into the office.
Yeah, we can do that. We could do that now. Yes. So that's we could do now, right? So as soon as that would come on the market, there will people that will want this, right? They will do it, and they've got like, I'll have some, right? So this is what I see is that as soon as something is possible, there will be people that will want it, and then if it's kind of catches on, then all of a sudden everybody will want it, right? It'll be an interesting future, right? We talked about diversity, right? And about how we don't want to become homogeneous, right? I mean, maybe this is something how in 150 years or 200 years, how our society is going to look like if we don't blow ourselves up or we don't bugger up the environment. Maybe in the future there will be these beautiful diversity of 2 meter and 1 meter tall humans that are pink and red, with gills, without gills,
like Star
with
Wars.
feathers, without
Star Trek.
Just
Yeah.
all of the diversity that you can think of. Everybody can be happy and can do whatever they want, but it'll be so diverse, right? I kind of love that idea. I think that would be really cool. That would be really cool. At least you will not get people go like, well, you're too fat. Go like, all engineered, mate.
Are they real? Ernst Wolvetang, thank you so much. I cannot wait for the future.
Okay.
If you want to be found, where can people go to find you?
Well, at the moment, they can find me at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. My email is there, e.wolvetang@uq.edu.au. It's all public. Drop me an email if people have a question.
Thank you so much.
Cheers.